Dr. Stella Mitrani-Rosenbaum, who I think a lot of you are familiar with her work as a pioneer with GNE Myopathy. And she joins us live from Jerusalem, Israel. Stella, I will turn it over to you, and I will see you all after the presentation. Thanks so much for joining us.
Thank you all, welcome, and thanks for joining us. So today I want to talk to you on a few issues that we are working on. First of all, I want to show you that GNE has specific roles in muscle that was not really clear till now. And then we will share the work we are doing to develop animal models that are essential to evaluate any treatment. So we will talk about muscle conditional mouse and also the development of GNE knockout zebrafish. And for the last years, this work has been very well supported by NDF grants. So we are really grateful for that. Okay, so when I first began working with GNE myopathy, then called HIBM, the flow of the research to solve this issue was very clear in my head. First, we needed to find the gene. Then we had to learn its function and some pathological mechanism that will lead to the disease, so we could target those mechanisms to design a suitable therapy. So indeed, we have found the gene GNE, and very quickly it turned out that this disease is in fact worldwide. And today, as you know, more than 200 different mutations have been found in the gene. So the function of the gene, GNE, was also very clear. It was known already. GNE is the key enzyme in the biosynthesis pathway of sialic acid. So everything seemed quite simple and clear. We had a gene that has a clear function, so there will be no problem to fix that. Except for some facts. One is essentially that sialic acid is in fact present in all tissues of the organism. So it was not clear whether why, in fact, only muscles would be affected. Okay. In any case, when looking at patients' muscles, it turned out that some have a reduced sialic acid level, but some do not or have a very slight reduction. And further, it is not clear how the reduction of sialic acid indeed results in the muscle pathology. So today, in fact, the role of GNE and of sialic acid is very clear and well known in brain development, but nothing is known to date of the GNE role in muscle tissue. So in the last several years, we decided to explore the GNE functions in muscle tissue. We did that in various systems, one in vitro system, but we will not discuss it today at all, but also in cells and also in vivo in entire organism. So at the cell level, what we can see that if we look at muscle patient cells which have mutated GNE in them, we can see only very mild changes by different studies by us and others, by transcriptomics and proteomics, for example. So we thought that we could see more dramatic effects of GNE if we looked at muscle cells that had no GNE at all, that is in GNE knockout cells. And those cells, one of those cells, were available as the embryonic stem cells that we have from GNE knockout mice. As you know, those mice are not born, but we can have the embryos and have the knockout cells. So that's what we did. And we know how to differentiate embryonic stem cells to any tissue in the organism, and in particular cardiomyocytes and skeletal muscle. That was important to us. So that's what we did. We took those embryonic stem cells, differentiate them in cardiomyocytes and skeletal muscle, and just look at them. So here we have wild-type cardiac muscle who have differentiated for 12 days, 20 days, and 29 days, and the same for the GNE knockout cells, stem cells, that differentiated at 12 days, 20 days, and 29 days. So if we look at what happens in culture, those cells are in culture, we can see that those cells can contract very well. You can see the contractions of the cells, can you? Hopefully. And if we look at the GNE knockout cells, the same, the cells can also contract and beat very well at the same pace, more or less. If we go at 20 days of differentiation and look at the contractions, we can see the wild-type cells going very well. And here, the knockout cells seem to be a little slower than before. You can see here some contractions. And then we go further to 29 days after differentiation of cardiac muscle, and we can see here again the contraction of the wild-type cells very well, very regular, and here the knockout cells that are very, very slow now. You can see maybe it takes a little bit of time to contract. Can you see it right now? Okay, so these cells indeed seem to be a little tired. And if we quantify those numbers, we can see indeed that at 12 days of differentiation, we have a similar beat rate of both the wild type and the knockout cells. After 20 days of differentiation, the beating rate of the knockout cells dropped to half, more or less, compared to the wild type cells. And at 29 days after differentiation, we see that the beating rate of the knockout cells is very, very slow. So now if we look at muscle, skeletal muscle cell differentiation, we used here a marker of differentiation, myosin heavy chains, to stain the cells. Only myocytes, that is satellite cells that have already differentiated to some steps and become now full skeletal muscle cells, myocytes, will stain with this marker myosin heavy chain. So here we have the wild type cells and the GNE knockout cells, and we can see that at 12 days after differentiation, most of the wild type cells are already myocytes, but we can barely see any cell in this field of the knockout cells that have differentiated to myocytes. After 20 days, the wild type cells form already a long structure. And here at the knockout cells, we can see myocytes, but they look like more single cells. And after 30 days of differentiation, we can see that the the wild type cells have already fused one to one another and form a fiber already, or the beginning of a fiber. In contrast, the knockout GNE cells are only single myocytes that are only beginning to differentiate. So in conclusion, we can say that GNE knockout really impairs muscle cell differentiation. So I hope I have convinced you that GNE now has a specific role in muscle. This is in fact the first time we can see a specific role of GNE in muscle tissue at very first levels of differentiation, but still the precise mechanism by which mutations in GNE lead to muscular pathogenesis and to the disease is still unknown. So a few years ago, we decided to take a more comprehensive approach for treatment, and we thought that gene therapy has a very strong potential, since the strategy is to introduce a wild-type copy of the gene, since the problem is not the presence of the mutation, but rather the absence of the wild-type gene. So AAV vectors based strategies are now very well established, and what we want to achieve with them is, in fact, the need of one single vector administration of the drug to get therapeutic levels of transgene expression that will be enough and that will be efficient for a long-term period of time, that is for lifelong if possible. So indeed, we have achieved some of those tasks. We have generated a viral vector that is a robust tool, since it can infect muscle cells when injected to healthy mice. It also allows the constant expression of the wild-type GNE for a long period of time, at least a year in the mouse muscle tissue. In addition, we could see no side effects at all. No toxicity could be detected in any organ of the mouse. And the vector is also immunologically well tolerated. But we don't have achieved one thing. And what we didn't show is the in vivo efficacy of this vector, because we couldn't inject the vector in GNE myopathy animal models, because simply we do not have those models. What indeed do we have? So we have GNE knockout mice, but in fact we don't have them because they do not survive past the embryonic stage, so they are not born at all. So we cannot use them at all. Now, when we have also knock-in mice that carry the mutation, the very frequent mutation in the Middle East or in Japan, what happens to those mice? Those mice, in fact, die within very few days after birth from severe kidney disease. In fact, we have some of those mice that survive and live normally, but they do not develop neither a kidney nor a muscle phenotype. So we don't have a model of muscle disease. There is another model of GNE myopathy. This is a model carrying a transgene, or a few copies of the transgene carrying the mutation that is frequent in Japan. But unfortunately, this model have no consistent phenotype and cannot really be used as a model of the disease. So in fact, why do we need animal models? Okay. So first, we need animal models to learn about the GNE function at the organism level. Okay. That could be somewhat different than at the cellular level. And by this, we could target a specific pathway that could be used for treatment, for example. Secondly, we need an animal model to screen the existing or novel drugs or treatments for identification of a potential treatment that can alleviate or prevent symptoms. So indeed, these animal models are really the last step for showing efficiency of any treatment that we can think of. So for the last few years, and with the support of NDF, we are working on the establishment of animal models that can allow us to explore the role of GNE in muscle. So first of all, we are trying to have mouse models. Again, the mice carrying GNE myopathy mutations do not show muscle pathology at all. And the knockout mice do not survive are not born, in fact. So we cannot learn anything on the long-term effect of GNE on muscle. So how can we overcome this problem? So the thing is, if we could generate mice that are knockout in the GNE gene, but not in all the organism that prevents them to be born, but only in muscle tissue. Okay, so this would allow us to look at the effect of GNE knockout in muscle tissue only. So how can we do that? So we can take advantage of a genome editing system, CRE-LOXP. This is a very well-established, for many years now, system to edit DNA. What it consists of, we know that there are specific short sequences that are called LOXP, that if we can find them in the DNA, in the genome, we can use a recombinase, an enzyme that can identify them, recognize them, and then cleave them, so taking out all the sequence fragments that are in between them. Okay? So for example, if we could introduce LOXP sequences around the GNE gene, we could use then this recombinase, this enzyme, to cleave out the LOXP sequences and the GNE gene with them. So we would get, again, a knockout of GNE. But this will be not different of the knockouts we have already, that we have obtained with other methods. So what should we do? Okay, so if we look at this recombinase, this is a gene, and as any other gene, the gene is expressed only by the activation of a promoter, a kind of ignition key of the gene. So many of those promoters are specific for certain tissues and will be active and activate the gene only in certain tissues. For example, if we have hemoglobin, hemoglobin gene is present in all tissues of the organism, but its promoter, it's only active in blood cells. So that's why we have hemoglobin produced only in blood cells. So if we could engineer a mouse that had a CRE gene with a promoter that will be specific to muscle, we could activate this recombinase only on muscle tissue. Okay? So in fact, what we would have, we could have a GNE with the LOXP sequences flanking it. We would cross this mouse with a mouse having the CRE gene led by a muscle-specific promoter. So the pups that we would have would have both genes, the CRE gene that will be active only in muscle, and the GNE gene flanked by LOXP sequences. So what will happen is that only in muscle, the CRE enzyme would be active, would recognize the LOXP sequences and take out the GNE. So we will have a GNE knockout mouse only in muscles. So this would allow us to follow up the effect of the lack of GNE in the muscles at different times. But we could have another problem. Let's say that the knockout of GNE in muscle would also be embryonically lethal, and the mice will not be delivered. So that would be a problem, right? So here we can use another modification of the CRE gene that will allow us to overcome this problem. We can use, instead of using the wild-type CRE gene, we can use a modified CRE gene that prevents the entrance of the protein of this enzyme to the nucleus where the DNA is in the cells. Okay, so we will have here now a mouse that will have the enzyme produced, but the enzyme will not get to the LogSP sequences, so the GNE will be there. Okay? So this mice will be born, and then when we would like to do it, we will inject to those mice a drug, tamoxifen, that will allow now the recombinase protein to go into the nucleus, to recognize the LogSP sequences and cleave out the whole fragment. So having a GNE knockout mouse only in muscle, and also we could control now when we would like to have this gene knocked out in the muscle. So this will allow us to look at the muscle features at any time point after development when we would like to do so. So again, to have all these things done, we need also to be sure that we can verify that indeed the GNE is knocked out in the muscle when we intend to do it. So as you may know, we still don't have a good antibody to recognize GNE. So before we go to this specific project to introduce the LogSP sequences around the GNE gene, we thought to generate a mouse where the GNE protein will be tagged by a small tag molecule that will allow us to identify when GNE is present or absent. So by CRISPR-Cas, we have indeed introduced at the very end of the GNE protein a small tag that's called flag that is in fact fused with the GNE protein. So we now have an antibody that can recognize this flag, and with it the whole GNE protein. And we have generated such a mouse. Here we can see a Western blot where we can see with the anti-flag antibody the GNE gene in many organs of this mouse, in the liver, in the kidney, in the spleen, in the heart is very, very faint indeed. But when we look at the muscles of this mouse, we can see here the gastrocnemius and the quadriceps of an heterozygote flag GNE mouse, and here the gastrocnemius and quadriceps of homozygote flag GNE. So now that we have this flag mouse, that we can follow up the presence or absence of GNE, we can use it to introduce the LogSP sequences around the GNE gene. Okay, so that's what we are doing. We have used again CRISPR-Cas to introduce those two sequences, and indeed here is an example of the clones that we got with the LogSP site at the 5' end of the gene and the LogSP site at the 3' end of the gene. Of course, these sequences have been verified, but what we haven't yet obtained is one mouse that has both the LogSP at this side and the LogSP at this side. Okay, so we are still looking for them, but at least we have a mouse that have the LogSP at one side. So in the meantime, we are looking for the both sides. Again, we have expanded this search colony with the LogSP sequences at one side, and we had to expand it because we need to have pregnant mice to obtain at least 400 embryos that would be injected now with all the CRISPR-Cas machinery to incorporate the second LOXP sequences at the GNE gene. So these manipulated embryos will be pre-implanted to foster mothers, and we expect to have delivery of at least 8 embryos. Normally we get something like 20% of embryos that are born from the ones that are pre-implanted. And of course we have to genotype them to see if we can really find the second LOXP sequence in those mice. So when we'll have that, hopefully very soon, we will cross them with the mouse which has the Gne modified gene with a muscle-specific promoter. And very fortunately, this mice is commercially available, so we will not have to do it. And then we will be able to generate a mouse with a conditional GNE knockout only in muscle and when we will want to do it. So these mice will be, of course, characterized for the phenotype, muscle structure, muscle function, locomotion and force, and also by more high throughput studies like transcriptomics and proteomics, so we can indeed identify some biological or molecular process that is altered by the lack of GNE. Once identified, this process could be screened for efficacy of any treatment that we can decide of. Okay, so now we have also, in parallel, we are also developing a zebrafish model for GNE myopathy. Why zebrafish? Zebrafish are vertebrates, and they have a good advantage that they have external fertilization, so we can collect the eggs that are laid by the fish and easily manipulated them. In addition, the major organs of those fish have similar function as in humans, and specifically muscle fibers development is very similar to humans. So to look at the GNE function in zebrafish, we wanted first to knock down gene expression because this can be done quite easily. This is done by injection of sequences that specifically can inhibit the GNE expression, RNA and protein, but this is only temporarily. It could last something like 7-8 days, okay? So we can do that very easily by taking the one cell stage embryos, putting them in a strip of agarose and injecting whatever we want. Here the inhibitor of GNE, and we can do that quite rapidly. We inject something like 200 to 300 embryos by session, and then we look at the phenotype of those injected fish. So after 4 days, we can see that the phenotype has changed. We can see, in fact, 3 kinds of phenotype: a mild phenotype, where we can see that the tail is a little shorter, an intermediate phenotype, where we can see, frankly, a tail that is completely curved, that is not straight anymore, and a severe phenotype, where we can see a very severe defect of development. The fish is not developing very well. So if we look more carefully at the muscles of those fish, we can see here normal fish, white-type fish, a very regular and homogeneous distribution of the muscle fibers. Okay, but if we look at the GNE knockdown fish. This is after 2 days, 48 hours of the middle intermediate phenotype fish. We can see a clear disruption of the regularity of the fibers. They are quite separate, and it even seems that there are less fibers than in the wild type. And indeed, if we quantify those fibers, we can see indeed that the fish that have been injected with the GNE morpholino have less fibers, and more significantly, if we inject also again a messenger RNA of GNE, we can rescue this the muscle fiber number. Okay? So then we wanted to see if the fiber disruption affects muscle function. To do that, we place each of the embryos in one well, in a 48 well plate. We put the plate in an etovision device that can monitor the movement of the larva, like this. Okay, so each well is monitored and all the data is collected and then analyzed. And indeed, after analysis, we can see that the fish that were inhibited by morpholino to GNE are much slower than wild type fish. And again, we can rescue this pattern by injecting again messenger RNA of GNE, meaning that this effect is specific for GNE. So we can see very clearly that GNE affects zebrafish muscle, but as I told you, this knockdown of GNE was very temporarily, very short in time. So we thought that we have to permanently knock down GNE in those fish. Again, we did that by CRISPR-Cas, and we got many lineages. One of them has a 4 base pair deletion at the very beginning of the GNE gene, and then it results in a stop codon, that meaning that the protein is not synthesized, so we have no GNE protein in those fish. And what we can see is that those fish live up to 8 days, and now we are looking at the muscle phenotype and muscle function of this fish. Again, we look at the structure, the function, and we have also begun an analysis of transcriptomics, again, to identify some biological or molecular processes that are more specific to the knockout that could be then screened for potential drugs. So indeed, here we have some preliminary results, and we can see clearly that the knockout fish have the gene expression in them is very different from the wild type or the heterozygote fish. And here we can see that many genes are down regulated, or many are upregulated of those fish compared to the wild type. So we are now trying to find a pattern in those fish that could point to a specific molecular pathway involved with GNE. Then when we identify this specific pattern, we can screen potential drugs or treatments to rescue the phenotype or that pattern that we have identified. So we will be able to monitor the restoration of the phenotype. So in conclusion, I want to say that animal models are really necessary for phenotypic screens of potential drugs. So we are trying to develop zebrafish or mouse models, so we can really try to monitor the treatments and can lead us finally to clinical trials. Okay, so I want now to thank you all my team, my present team. These are my present lab members, Lena, Miri, Esther, Avi, and Chagai, and a long list also of former members of my laboratory. And I want to thank my long live collaborators, Zohar Argov, Hannah Ben-Bastat, Yoav Gotthilf, Rebecca Hafner, and of course the NDF for its great support. Thank you very much.
Thank you. Excuse me. Thank you, Stella, so much for that really informative and interesting presentation. We're going to move to our question and answer part of this event, and I would like to introduce Dr. Carmen Bertoni, who is NDF Scientific Director. She's having a little trouble with her camera today, but she's going to go ahead and moderate the Q&A section of this for all of you. So please feel free to enter your questions. I see there's quite a few here, Carmen. And so Carmen is going to go ahead and ask your questions to Stella, and I'll be back just to thank everyone.
Good morning, everybody, and thank you, Stella, for the beautiful presentation. I'm going to go straight to the question that people have logged in, and also I have a few questions on my own as well. Mangesh is asking, what is the difference between a knock-in and a transgene mouse model? Can you explain in lay terms to us what are those differences?
Yes, I will try. Okay, so a knock-in mouse or model is when you replace the gene with the modification you want. So for our purpose, the knock-in mice have the mutation we want instead of the normal gene. So they recapitulate exactly the genotype of the patient who has this specific mutation. This is the knock-in. Now, a transgene is when you add a gene into the cell, but you don't really care where this gene goes, okay? So you have the gene, for our purpose, the GNE gene, that is added to the cell, to the genome, but it can be anywhere. It's probably not at the place where it should be. We can also have many copies of the gene that we don't have normally. Normally, we have 2 copies of the gene. So here, when you have a transgene, we can have 2, 3, 5, 10, 20 copies, okay, of the gene. So there is an obvious difference, because then this transgene, it depends where it gets in. It can affect other genes. So we don't really have control of it. Did that answer the question?
I think it's clear, at least for me. I have a few personal questions here. Why do you think it is so difficult, or that's been so difficult in the past, to generate a mouse model? And this will be question number 1 about the mouse model. The second is, do you see differences in phenotype among the different knock-in mice, depending on which mutation you are pulling in, whether it's the M743 or the V603?
Okay, so I begin from the second question, if it's possible. So we have worked with the M743 T modification, but from the literature, I can see that both mutations gave the same phenotype, that is the kidney phenotype, okay? Now in our case, we have managed to have a colony of mice that did not die from kidney. They were living very well. They had a normal lifespan. We hoped to see their muscle phenotype, but we didn't. So, but that's maybe the exception, okay? So I guess today we know of 2 knock-in mice, the one with the M743T mutation and the one with the 602 mutation. And as I can see from the literature, because the second one has been done by a Japanese group, not from Dr. Nishino, another group that we don't really have contact with. So the literature describes the same phenotype, roughly. So that's for the mutations. Then about what you are asking, why it is so difficult to have a mouse model. Well, many times you don't recapitulate a phenotype from human in mice. Of course, many times it's close or very close. In fact, even with DMD mice, the phenotype is not recapitulating exactly the DMD disease in humans, but you can still have some points that you can look at. Here, we really cannot see anything in muscle with the classical models or the classical techniques to have the models. It could be maybe because let's say that GNE affects sialic acid. For sure it does. Okay, we know that GNE mutation affects sialic acid. We don't know if sialic acid really leads to the disease, but we know that GNE affects sialic acid. So maybe the loss of sialic acid is quite important and masks the other phenotypes that we would want to look at that are milder for the muscle. So that's why I think specific knockout in muscle maybe could answer better the question.
Okay, thank you. And I do see some questions that are similar. I'm going to try to put the question in a logical flow so that we don't jump from one argument to the other. It's actually Nancy is asking a very specific question on the mouse model. Do we have a timeline for the mouse model? Or when do you think the mouse model would be able to be generated? I know this is a difficult question when you start in science, you can never predict
you can never predict
specific numbers. But if you can give some kind of clues of where you think you are right now and a possible timeline for the future.
Yeah, well, as I showed you, we have, let's see, half a mice model, if I can say. That means we have 1 LOXP close to the GNE gene. Now we are working on the second one. So that could take a few months, okay, to have the whole, because as I said, we have to expand very much this flagged LOXP mouse. This would take some months. Then when we have it, we can cross it with the commercially available mouse. So that could be quite quick, I guess. And then when we'll have the second generation of those perfect mice, if I can say, with the LOXP and the CRE gene in them, we have to begin to inject tamoxifen to see how the knockout of GNE affects muscle. So, of course, I guess something longer than a year or year and a half, I will hope. If we have it in a year and a half, I will be very happy, let's say.
Great, that's very promising. We have a question from Tara again on the knockout mouse, and then we move to the question for the morpholino, for the zebrafish. Tara is asking, will the AAV vector work on your knockout mouse, or are you planning to use CRISPR-Cas9 gene editing technology to correct the gene? If you plan to use CRISPR-Cas9, is this going to be done in collaboration with the LEC lab?
Okay, well, I think that for gene therapy, or this specific problem we have, I think the addition of the GNE wild type would be better. Why better? I can explain that. I mean, the correction of the gene is essential when the problem is when the gene is mutated, okay? But as I said before, we think we have a disease not because the GNE is mutated, but because we don't have any wild-type GNE. And this we can say because if we can see at heterozygote people, we see they have one copy of the mutated gene and one copy of the wild-type gene, and they are perfectly well, okay? So the problem is not the mutated gene, the problem is the absence of the wild-type gene. Now, of course, we can correct the genes, but we have to look here very carefully at efficiency, because for any method we use, or supplying the entire gene, or supplying all the machinery of the CRISPR-Cas to fix the mutation, we have to deliver that with a vector into the muscle cells, right? So this delivery, today at least, is best done with AAV vectors, okay? So if we use the whole gene in AAV vectors, all the process is one step. We have to infect the cells with AAV GNE vectors. Once the GNE is in the cell, it begins to work, hopefully, and everything is fine. Of course, the efficiency is far from 100%. So we will not have AAV GNE vectors in all the muscle fibers, okay? We will have much less than that. Now, if we use the CRISPR-Cas to fix the mutation, here we have two steps, not one step, but two steps. We have first to deliver the entire machinery to the cells with the AAV vectors. So that, again, is not 100%. So we have the same efficiency of delivery, okay? But then the CRISPR-Cas itself has also not 100% efficiency. So here we will just lower the efficiency of the potential fixing of the mutation. So I think that it would be better, more efficient to inject AAV with the whole gene. But of course, when we get at this point, we can discuss everything with everybody, and if we do change our minds, of course, it will be done with the LEC lab, that's for sure. Yeah. I don't see any other way to do it anyway. Yeah.
Absolutely. And I have another question here that is very basic. Can you explain in lay term why do we need the animal models? And if we cannot succeed in generating the animal models, will we be able to go into gene therapy or any other therapeutic approach without a model, whether it's a mouse or it's a zebrafish?
Okay, so again, there are many steps to go from an hypothesis to a treatment to the clinical trial, okay? Many of them. Okay, so one of them is preclinical studies. So we have to show that the treatment we suggest is safe, first of all, so that we have already show in mice, of course. Then we have to show that the basic hypothesis that we are working with works. That is, we have shown that GNE indeed is produced in the cells that have been injected. But then we have to show that the treatment also address the problem, that is efficient to treat the problem. Okay, so this we cannot show if we have not an animal model to show it. Okay, because if the animal model does not present any muscle problem, we cannot show by any means that the treatment we suggest will fix the problem, because we have no problem in those animals. Okay, so this will be really difficult for convincing any regulatory agency that the treatment is efficient, any treatment that we show. On the other hand, if we really show that there is no animal model, not because we didn't try to do it, but because it really doesn't work, I believe that it could be still done, because there are other examples of diseases where there were no good animal models that the FDA at least have given permission to go ahead with clinical trials. So I think it is an essential point to try very thoroughly and methodically to have a muscle phenotype that we can treat in an animal. But I believe that if we don't have it, we still will be able to go to clinical trials.
Thank you, Stella. That was a great answer. We're going to move to the question that's specifically related to the zebrafish model. And Nancy has a basic question. Do we have a zebrafish model now?
We have now a knockout zebrafish that lives for around 8-10 days. We have. So that means, in contrast of what happened with the mouse that are dying before they are born, so we cannot work with them, here the knockout zebrafish is in our hands from day 0, okay? So we have 8 days to try to find a treatment that will in fact overcome the death of the zebrafish, okay? So we can use them to monitor any treatment we want to, okay? Is the answer clear?
Yes, I think that's an excellent
I cannot hear you very
answer.
well. Okay.
We might be having some trouble with Carmen's audio.
I cannot hear
Getting messages from my computer at least that yes. Can you hear me?
Yes.
If please, if anybody has their mic on, please mute yourself because it's slowing down the entire connection. We have a technical question from Noah, Dr. Westlader. You guys have had the chance to hear his talk last week. It was very insightful. So the question from Noah is, with the morpholino knockdown in the zebrafish, was there a change in the size, what we call in science a cross-sectional area of the muscle fiber, or just in the number of muscle fibers?
Well, we saw clearly a change in the number of the muscle fibers. I'm not sure we have done a cross-section. I'm not sure of that. I have to check, and I'll give you the correct answer. But I don't think we have looked at that carefully.
Okay, and again from Noah, the changes in the muscle cell markers expression in cells that are differentiated were quite striking. Did you stain for any extracellular components like collagen, fibronectin, or membrane proteins, sarcoglycan, dystroglycan, laminin, etc.? This is a more technical question.
Yes, well, we did indeed stain for many markers, but I don't have them in mind. It was a few years back, sorry. I have to check exactly what, but yes, the conclusion was that all the differentiation is delayed. So I can get, again, the whole thing and get back to you, Noah. Sorry.
No problem. This is a question from Catherine. In the zebrafish morpholino knockdown, is the degree of phenotypic severity correlated with the level of knockdown? Do you see the greatest reduction in GNE mRNA and protein expression in the most severe phenotypic classification?
Yes, well, we don't have protein expression, but we had messenger RNA expression, and it seemed to be less, yes. I mean, the less expression, the more severe the phenotype.
Okay, great. We have one more question from Mangesh. We are going back to the AAV-based gene therapy approach. Is this approach safe and effective in that the gene expression can be seen for up to a year or more? Since muscle fiber have longer lifetime, should we expect a longer gene expression with gene therapy for GNE myopathy?
Well, the 1 year long expression was in mouse, okay? So if mouse live for, let's say, 2-3 years, so that's a long time, okay? Now, from other systems, other diseases, like the disease of a blood disease, okay, where that has been treated with AAV. Patients have been seen with the presence of factor VIII for 10 years now. So, of course, this is in blood cells, okay, and not muscle cells. But again, blood cells are really much shorter lived, so they can express the hemoglobin for so many years. So that means the AAV vector is still there. Okay, so we expect that in muscle cells, once the vector reaches muscle cells, it will stay for a very long time there, stay active for a very long time there.
Yes, that's. I think this is one of the beauty of working with muscle cells. Is the slow turnaround of the muscle fiber. We have one question that came in through a text, and given what has been done right now, is there anything that can be done more to speed up the timeline for animal models?
Well, animal models are animal models. That means the animal has its biology, its reproductive time, and I am afraid we really cannot do anything for the methodologies we have chosen to, okay? So we are limited to the biology of the animal we choose, okay? So in fish, it's better, it's easier, it's more rapid. In mouse, it's much slower, and I think we have to live with that. I don't have any methodology to bypass that. If there are, it will be welcome.
Yes, that's exactly how everybody is feeling. But I think in terms of what groups like yours have done in the past, have really shown you guys are really working at it, and we are exhausting all the possibilities. So we should be able to get the animal model, and if not, as you explained, there are ways to overcome, but we still need to keep trying at
Right.
our best. We have another question from Mangesh. If AAV gene therapy is used to deliver the entire wild-type GNE gene to muscle cells, does it need to be located anywhere close to the original DNA molecule for full expression of the GNE gene?
Not sure I understood correctly, but if I understood, you mean if the GNE gene has to be at proximity of the GNE gene in the genome? I don't think this is important, because what we need from the AAV GNE vector is the protein, okay? Is the GNE protein, not the gene. And the protein is in the entire cell. So it doesn't matter where the AAV vector is in the cell. Anyway, the AAV vector stays independent of the genome in the nucleus of the cell. So it's at proximity of the genome, but it's not into the genome. So the protein works well in all the cells. I don't think it's important at all. Yeah.
Right. So in other words, it just needs to be in the nucleus of the
Right.
cells. And one of the beauty of the AAV system is the fact that it does not integrate. So it can be separate from where all the DNA is contained. And I hope this answers Mangesh question. There are no more questions that I can see. Oh, wait a second. There is another question, is if mouse model work, how long would the gene therapy itself take?
Well, this is a difficult question. I don't know. Well, if the muscle, first we have to see that the mouse model works. Then we have to do the treatment to those mice to see if the treatment works. And then we have to go to the FDA with that. And then I don't know what the FDA will have. So we have to have the model, and then to see that the model works, and then to see that the treatment works on the model. So that could be quite a while. Yeah, I don't know. Difficult for me to say. Yes.
Okay. So scrolling down, and Tara is asking, I know you don't have a crystal ball. However, would you please say what would be the best case scenario for GNE myopathy patients?
Well, the best scenario for me would be that sialic acid would work. That would be the easiest. Okay. So then after that, what we have to see if we have a good model that the treatment, gene therapy treatment, will work on this model, and that the FDA will really accept and give its green light to go ahead. That's, in my view, the best possible scenario to date that we can expect to have at the moment.
Okay, and that was the last question. I want to thank Stella for the insightful presentation and for the beautiful work that they have been done over the year. And with that, I'll turn it over to Nancy. And thank you very much, everybody.
Thank you, Carmen, and thank you to Carmen for so eloquently moderating everyone's questions. Hopefully everyone feels heard and got their questions answered. Of course, we're always available via email or through our website for questions that you have following up to this. And as I mentioned earlier, the presentation will live on our video library on our website as well. We'll have that up as soon as we can. Stella, I really want to thank you for taking your time and energy.
Thank you very much.
These presentations take a lot of attention, and we so appreciate your giving us part of your Friday night. I want to thank everyone for joining us. We had a great cross-section of people participating today, patients, some of our own certified patient advocates, scientists, industry leaders, family members from all over the world. So I'm so glad that this pivot to a virtual symposium has worked, given everything that all of us have going on in the world. I have a special message from Lale, who is currently en route in her car. She wanted to thank everyone for participating and tell everyone that you made the soundtrack for her drive much more interesting. And I also just want to remind everyone that next week we've got David Clements from NeuroNext Network at Massachusetts General Hospital joining us to update everyone about NIH's NANAC trial. That'll be next Friday, June 12th at our regular time, 12 noon Pacific. And if you have any comments or questions, obviously feel free to reach out to any of us at NDF. Stella, thank you again for your time.
Thank you very much to you.
I hope that everyone stays safe and stays healthy and stays in touch. Thank you.